Okay, so today I noticed when I was drawing the rough ER on the board, saw a few faces that wanted me to say, hey, maybe I should go back and discuss this a little bit. All right, so what have I drawn here? I have drawn the outside surface of the rough endoplasmic reticulum. Now this orange part right here is a ribosome. All right. And with this ribosome, this is going to convert the mRNA shown on the left into protein. Now this mRNA comes from the nucleus, and it's akin to a file off of your computer. Okay, your computer has thousands and thousands of files and documents on it, right? Well, this mRNA is a copy of a single document or a single picture file. Now, this comes from the nucleus and goes outside of the nucleus, through those pores we talked about, to the outside of the cell, and now we are now outside of the rough endoplasmic reticulum. Now, this is on the inside of the rough ER. And what's going to happen is this mRNA is going to interact with the ribosome, and as it interacts with the ribosome, it will produce protein inside of the rough endoplasmic reticulum. Okay, so now we have the final product. Before you saw the mRNA was here, right? It was over here. And now it has moved to here. And notice what's happening. The mRNA is interacting with this ribosome. And as it interacts with the ribosome on the outside, it is producing a strand of protein. And these are, remember from previous discussions, these are each amino acids. It is making a strand of amino acids inside of our rough endoplasmic reticulum. Again, we are making protein inside of our rough ER, or our rough endoplasmic reticulum, because mRNA from the nucleus is sitting on the outside and moving through the ribosome, shown in yellow here, and as it moves through the ribosome, it makes the amino acids come out the other side inside of the rough endoplasmic reticulum. Now, how a ribosome works, how mRNA works, and DNA, and these amino acids, we're going to discuss this for the next test when we discuss the central dogma of molecular biology. But for now, let's just leave it as mRNA is on the outside of the rough ER, and this mRNA interacts with the ribosomes, and the ribosomes convert the mRNA into a sequence of amino acids that we know as a protein. And this goes inside of the rough endoplasmic reticulum. And again, the rough endoplasmic reticulum is a membrane, and that's why we see that phospholipid bilayer here. So that's why I've drawn the red and the blue, indicating the hydrophilic blue and hydrophobic tails. Now, when we look on the outside of the ER, we're going to see these ribosomes, and when you look at it with a microscope, you're going to see little marks covering the rough ER. That's why we call it rough ER, because it has these ribosomes on the exterior. And then go back to the function here. The ribosomes convert the mRNA to a strand of proteins inside of the rough endoplasmic reticulum. And these proteins then can be processed and things can be done with them. So that kind of gives you an idea of what's happening on the surface of the rough endoplasmic reticulum. And again, to reiterate, the rough ER produces protein. mRNA sits on the outside, moves through the ribosome, and as it moves through the ribosome, it makes a strand of protein on the inside. Or better yet, a strand of amino acids, known as a polypeptide, or a protein on the inside of the organelle.